Hi everyone, I'm going to tell you about this article called A Three-Dimensional Organotyping Microfluidic Model for Glioma Stem Cells, Vascular Interaction. This is about a PDMS microfluidic device to analyze the interaction between glioma stem cells, endothelial cells, and extracellular matrix using hydrogels with matrigel and fibrin. Let me show you the images of this interesting article. Then use here. You can see this one. The objective of this work is try to modulate or modeling the proliferation, invasion, and stem cells maintenance processes that are involved in the interaction of glioma stem cells and endothelial cells that can use with patient-derived cells from biopsies or maybe using established cell lines. Okay, here in this schematic image, you can see the design of the device that has a central chambers where GB3 cell lines that are glioblastoma cell lines that were cultured, a stroma layer with hydrogel to mimic an extracellular matrix, and a peripheral chamber that Hubeck cells are cultured. Hubeck cells are established cell line for modeling an endothelial cell. Fibrin in hydrogels were demonstrated that can activate vasculogenesis process that can be appreciated in these Hubeck cells. So in the next picture here, you can see that the Hubeck cells after 3 days of culture, their cells were able to fabricate microvessels that can be stained with dextran and CD31 that are used to characterize endothelial cells. Here you can see the microvessels and the staining with the CD31 and the API to stain the nuclei of these Hubeck cells. In the next picture, we will see that stem cell culture require specific conditions to keep the stems phenotype that can be guaranteed with a serum-free media or low amounts of serum culture media, and different growth factors can be added to this culture media. Here in this picture, you can see the stem cell markers expression by different GB3 cells in 3 different culture media. NSC supplemented with serum, and EGM2, that all of them are considered optimal to keep stem cell phenotypes. Here you can see the expression of nesting, GFP, SOX2, and CD44. All of them are pluripotency gene markers. But in making a comparison of 3 different culture media, no significant differences in the marker expression are registered. In the next picture, you will see that here, no, the next here. Here you can see that and they registered this culture media NSC. You can see that here they register the biggest extension of cell body, the nuclei per chain, and the nuclear in field of view. And the GBT cells are cultured with Hubeck cells. And this is Important because it can be explained for the activation of migration processes with chemicals um, that can be present in extracellular components. Okay, here you can see the extension of the GB3 cells when they are cultured with Hubeck cells in the different culture media. And the significant differences are registered in this culture media in all of the three parameters that they evaluate. Here in the next figure, they evaluate the proliferation of these cells inside the device using Ki65, Ki67 and YouTube proliferation kit, and they saw that no significant differences in proliferation were observed in tumor cells with Hubeck cells, but they demonstrate that these tumoral cells can proliferate in the devices. And here you can see that this Ki67 and the EDU staining to check the proliferation stage of these cells, but no significant differences are observed in all the conditions that they evaluate. Okay, in this article, the authors could optimize a microfluidic platform to modulate an invasion assay considering the interactions between tumor cells, endothelial cells, and the elements of the microenvironments. Thank you so much.